Thanks everyone for coming today. I'm excited to have our speaker today, Shella Keilholz, who's from Georgia Tech / Emory. It's a joint department, I believe. Um, she's going to talk with us about spatial temporal patterns of intrinsic brain activity. And she just returned from Australia, so thank you for coming despite the time change and everything. Shella Keilholz is an associate professor of biomedical engineering. She has a background in physics from Missouri University of Science and Technology and the University of Virginia. She did postdoctoral research on neuroimaging at the NIH, does a lot of work on dynamic functional connectivity, brain networks, and dynamics across states. And what I remember having a conversation with Shella at, I believe it was an NSF meeting a few years ago, and we were talking about spatial patterns of brain activity and traveling waves and things like that. And at that moment, I hadn't really considered the possibility that traveling waves could be the kind of signal that you could measure with effectively with fMRI signals. So I've kept an eye, and she told me that you could, and I've kept an eye on her research since then. So I'm really happy to have her here to talk about that perspective. Before I let Shella take the floor here, I just want to remind everybody that our Wave Club seminars are archived on YouTube, which is really cool. It's becoming a great resource with something like 15 or so different speakers online. The whole recordings are there. It's a nice record of this meeting going forward and a way of reviewing what's going on in the field. And also, if you want to, so this is the link here. You can just go to YouTube and you can search Wave Club. And you can find our recordings from right now. We also have an email. If you want to be notified of upcoming meetings, we meet about once a month. You can find us on Blue Sky, where you just search Wave Club Seminar. I think we might also be present on Twitter as well. And we have an email list as well that you can see on all of these places, and just subscribe to our email list if you want to attend and learn about future meetings. The plan for the meeting is that Wave Club is usually an informal session. Shella will present for about 40 minutes, and then we like to keep the whole meeting to about an hour, with the remaining 20 minutes for questions. If you have little informal things, it's great to jump in, but the best way to frame bigger questions is to use the chat or the Q&A feature to type in your questions, and then Uma and Irfan will kind of help us keep track of the questions, and then hopefully we can bring you on stage virtually to have a conversation about your question at the end. This is just a way of helping us organize things. And then finally, we're always looking for presenters at Wave Club. If you are interested in presenting at Wave Club in the future, please contact us. Self-nominations are super welcome. This is an exciting new field, and sometimes Bard and Uma and Irfan and I lose track of we like learning about new people doing work in the area, so please reach out if you're doing relevant work, so we can help get our presenters organized. Speaking of our presenters, we have the next couple sessions of Wave Club scheduled. On April 7th, Dominic Kohler from Berlin is going to be presenting on some work on brain stimulation and traveling waves. And on May 5th, Sage Chen, who's a professor at NYU School of Medicine, is going to be presenting as well. He does work on computational models of traveling waves related to theta oscillations and place cells and things like that. Okay, I'm going to stop sharing my screen because you didn't come here to hear me. Shella, why don't you share your screen and dive into your presentation? Thanks again for coming.
All right, thanks again for having me. Let me get everything going here.
And if anybody has questions, I really encourage you just to type your questions into the chat. We're not using the Q&A feature today, so just type them into the chat and we'll keep track of them and then organize the Q&A in a coherent fashion towards the end.
All right, can you see my slides okay?
Looks beautiful. Thanks, Shella.
Perfect. Well, first of all, I've got to say, you guys are really going to enjoy the talk from Dr. Dominic Kohler. I just heard him present in NUSA, and he's doing super cool stuff. So that's a teaser for next time. I'm excited to have the chance to come and tell you about some of our work today. And this is going to be more from a whole brain, very slow perspective based on things that we can measure with fMRI. So please do feel free to stop me if you have any questions as I go along, and I'm really looking forward to having some discussion afterwards. So if you look at the brain with fMRI, and even if we're not doing any task, we see a lot of fluctuations in activity over time. So over here, we've just got a video showing, deviations from mean activity in different areas, where warm colors mean higher than usual, cool colors mean lower than usual. And so you can see there are changes and that they vary in space and that they have certain patterns seem to persist, and you see kind of things that maybe look like networks. So this is going on all the time. And we know that this intrinsic brain activity is important because if you look at it from an energy perspective, the brain gets 20% of the body's energy, but your increases in baseline blood flow, for example, during task performance are only about 5%. So most of the energy seems to support this sort of intrinsic signaling. We know that these patterns are influential. If you have an incoming stimulus, it interacts with this kind of background activity, and that gives rise to some of the variability that we observe in samples from, one trial to another. And we know that these patterns are very persistent. And if we look at the spatial structure of these patterns, people have done studies where they tried looking at different conditions. And so here's a study showing that there are differences between wakefulness and deep sleep. However, it's true there are differences, but what I think is more striking is the similarities. So these patterns don't go away. Even when you're asleep, we see similar patterns in different species, so mice, rats, monkeys, under different conditions, from sleep to wakefulness to sedation to anesthesia. And so, these are a very fundamental property of the brain. And I want to point out that even when we're doing task-based fMRI, these patterns are still present. We have this intrinsic background going on in the activity going on in the background, no matter what else is going on. So I think, that's why we'd really like to understand what the deal with it is. So I don't know how much you all know about fMRI, but the kind of study that I just showed is called resting state fMRI. Not because the person is necessarily resting or sleeping, but just because we're not giving any specific task or stimulus. So you can also think of it as, an uncontrolled task. We put somebody in the scanner, we say, don't fall asleep, and we just acquire images that whole time to look at what's going on. And people have come up with so many ways to look at this data that I'm sure you've seen some before. So today I'm just going to talk about two: correlation matrices, which kind of tells you the overall time average structure of the activity. And then this feature called quasi-periodic patterns, which incorporates aspects of these traveling waves that I hope that you all will find of interest. So to go into the bit about functional connectivity, if you haven't seen a lot of this before, one of the ways that we like to summarize this intrinsic activity that we see in the brain is by doing correlation between time courses from different areas. So you can see up here, there are different atlases that we can use. They have different resolutions, different ways of defining areas. And, there's just one example shown up here. There are many, many atlases. But the idea is that you can take the brain and you can divide it into chunks. And then we can take the average time course from each chunk and do correlation between that time course and the time course from every other chunk. And that's how we come up with these correlation matrices that you see at the bottom. And they're organized to show that there are kind of these modular areas of correlation that we call functional networks. And based on those correlation matrices, people do things like go back and make graphs and talk about nodes and degree and efficiency and things like that. So these functional connectivity matrices are the basis for all of that. They're also the basis for a lot of the work on brain states. People will look at different patterns of functional connectivity over the course of a scan. Okay, so that's what I'm calling the time averaged approach. And I call it time averaged because when you typically make these functional connectivity matrices, you take the entire time course from the scan and do correlation of that. So for one 10-minute scan, you get one functional connectivity matrix. So it's basically averaging across those 10 minutes to summarize the common structure. The other method that I'm going to talk about is something that we came across. Well, we came up with this in my lab. It's not something we went looking for. And I think I'm going to tell you how we came across these patterns, because I think that's the only way it makes sense to understand this algorithm that we developed. So we were very interested in dynamics of the brain. And so I told one of my early students, I said, well, let's look at the data. And I had in my mind that maybe we'd look at how functional connectivity changed over time or something. But instead, he made a movie. And when he made the movie, we could see these waves of activity. This was actually in a rat, not in a human, but we could see these waves of activity moving from the lateral sides to the midline over and over, very repetitive. And we weren't sure what was going on with that, but we wanted to characterize it a little bit better. And so he made a pattern finding algorithm. And what we do is we take a randomly selected chunk of time. So it's a 3D volume by time. And then we do correlation between that and the whole time course from the whole scan to find other chunks that look like that little section. And then we average those sections together that we find where they have high correlation. And we make a new template, and we do this again until that template that we've pulled out repeats and converges. So what we end up with is this pattern template, which is again 3D by time. So it's a movie of activity over a volume. The time varies anywhere from about 5 seconds to 10 seconds in the mouse or rat to about 20 seconds in the human. And then we have this sliding window correlation vector that tells us when that pattern is particularly strong. And we called these patterns quasi-periodic patterns because they kind of showed up again and again in this repeated manner. So let me show you what that looks like in humans. Now, I told you we originally observed this in rats. And at first we thought we might be looking at an artifact of anesthesia or something. We weren't sure it was so interesting. But we put data from awake humans through the same algorithm, and out popped some patterns that were very familiar from previous work on functional connectivity. So I'm going to play this movie of the template now. But before I start it, what you're seeing right now is blue showing again lower than average activity, orange showing higher than average activity. And in case you don't look at human fMRI data very often, right now the blue network is the default mode network, and the orange network is the task positive network. So there's kind of this division of the brain into these two anti-correlated networks. So as I play the movie, you see that the patterns get stronger and then they switch, and that you do see some propagation along the cortex in between. So I'm going to play it again. Watch kind of along this medial slice. You can see some of that wave-like propagation. That's one of the places we see it. It's a little more obvious in the rat, I think, because the cortex isn't so folded, and so we get a little bit clearer picture of the propagation, but we do see it in the humans. And these are whole brain patterns. So I'm going to show you zoomed in on the subcortical areas. So these changes here occur in a time-block manner to the same changes that we just saw in the cortex. So this is truly this sort of whole brain integration of spatiotemporal patterns. Again, there's propagation in some of the subcortical areas. I think somewhere in here is a good one to look at, if I remember. Maybe it was the other one. But I'll play it a couple of times, and you can kind of see what catches your eye.
Shella, is this one person or is this an average?
This is an average. In fact, this particular video that I'm showing is an average of a lot of data because we use the HCP data. You can get it in a single person. It's a little less clean, and it depends a little bit on the data quality.
Thanks.
Okay, so that's kind of where we started looking at these spatiotemporal patterns of brain activity. Now, I'm going to use QPPs as a term more generally for any kind of repeated spatiotemporal pattern that covers the whole brain. And you're going to see why in a minute, because there are a few different algorithms that we've used and that others that have used to pull out these patterns now. And so there's a lot of different terminology in the field, and I don't want to call these things by about 4 different names. So we'll just call them QPPs. And in general, we see about 3 of these that define most of the variance in our human resting state fMRI data. So the one I just showed you is the DMN versus task positive network. And the reason I'm showing that one is because when we first started looking at this, we were always doing global signal regression prior to doing this type of analysis. And the first pattern we see actually is mostly about the global signal. The third one is a little bit more divided between sensory, motor, and visual regions, and we haven't done as much work with that one yet, so I'm not really going to focus on it today. So I mentioned you can do this with multiple algorithms, and one of the ones that we use is a complex PCA. The nice thing about this is you don't have to predetermine the window that you use to define the patterns. Instead, you do a Hilbert transform, take the PCA of the resulting complex data, and then you can pull out the primary components with a phase that gives us that same sort of propagation. So here, I don't have the movies this time, just the phase maps and the reconstructed time courses. So this is that one that is mostly global signal. So the primary component. Here's the same one I just showed you a while ago, which is more divided into default mode and task positive network. So if we don't do global signal regression, we get both of these, and then the other one that we are still not very clear on yet. So what I wanted to spend the rest of the time talking about is why we have these. Where do they come from? Because this is something we're really trying to understand. And of course, with resting state fMRI, we're always looking at hemodynamics. And we assume that these are linked to the underlying neural activity via neurovascular coupling. And that's true, but there's also evidence that that link is maybe not as tight as we would like it to be. And we also don't really know what organizes that spatial pattern of underlying neural activity. So all of this is a matter of interest to us. I'm not really going to talk about the vascular geometry component today. We can talk about that on discussion if anybody's interested. But for now, let's stick with the neural sources for these kind of large-scale organizations. So, one idea that we had is that, maybe it's just a resonant of this network structure. I showed you how you can take these functional connectivity matrices, turn them into graphs. You can think about intrinsic activity interacting over this graph and showing some kind of repeated pattern. So that's one idea we had. Another came from some of our neurophysiology colleagues who pointed out to us that there are these deep brain neuromodulatory nuclei that have very widespread projections. And at first, they didn't seem like good candidates because people said, well, they innervate the whole brain. But then, our colleague explained to us that they have receptors that have different levels of sensitivity. And so you could get this sort of spatial selectivity from deep brain nuclei like the locus coeruleus, the nucleus basalis and minor, maybe the RAF-A. And so we started to wonder if maybe those weren't involved in how these patterns come about. So I'm going to go into each of those possibilities a little bit here. And the first thing to note is that if you look at structural connectivity versus functional connectivity, it looks a little different. And what I mean by structural connectivity is the same kind of matrix that we talked about making for the functional connectivity before, but now instead of correlation between time courses, we're looking at the number of white matter streamlines between them detected with diffusion tensor imaging. And that has problems of its own and sensitivity gaps of its own. But what you can notice is that if you look at the structural connectivity matrix and the functional connectivity matrix, they have some similarities, but they're not at all identical. So it's not that we have, that functional connectivity reflects one-to-one coupling via a white matter track, which, seems very naive now. But when I started, it seemed like that's kind of what people thought sometimes. So then we came across these puzzling features. Like if you look at the caudate putamen, the caudate putamen is very strongly coupled. So you always get these nice bilateral functional connectivity maps for caudate putamen, but it's not directly connected. So we found that very confusing. Why would you have that without a direct connection? And, of course, people would say, well, it's part of the polysynaptic circuit. But if you start, going down that pathway, it seems like everything could be connected, right? So we went to this network modeling approach to get a better idea of kind of where those resonances might come from. So the way this works is we take that structural connectivity matrix, we build a graph framework. So we're building the graph from the structural connectivity. At each of these modes, we model neural activity in some way. And there are a lot of different neural mass models you can use. We use the Kuramoto model, the firing rate model, the Wilson-Cohen model. We get similar results with all of them. So we model the activity at each node with this equation that allows these interactions between nodes. We let that run to produce this sort of intrinsic background activity. And then we take what that spits out and put it through this QPP analysis. And when we do that, I'm going to have to explain how I'm showing this here, but when we do that, we get some features of QPPs. Okay, so to make this a little easier to see, since we've got it on a graph network, what we've plotted here is for each of the nodes, we've plotted whether it's above average or below average activity over the course of the template. And so in this plot at the upper left, we have condensed the activity that we see in that QPP template on the human brain into this 2D plot. So this is what the empirical QPP looks like. You can see that division into kind of positive and negative components that alternate, and you can see the sort of blurring along the side. That's the propagation, this pattern moving from one area to another. So when we look at the outputs from our brain network models, we do get this kind of division into two modes, and they kind of line up with default mode and task positive. And that's true whether we use the firing rate model or the Kuramoto model. But you see that the pattern is a lot less complex. And so we're not getting that same sort of propagation. It's more like this sort of standing oscillation. So it captures some of the features of what we see in the QPPs, but not all. Now, with the caveat, the model that we use does not actually include physical adjacency. It's all based on these white matter tracks. And so it doesn't actually allow activity to move from one node to the next node down the cortex. And so an improved model might give us a more realistic QPP. So I think we can't rule that out yet. Okay, but then to come back to these neuromodulatory systems, we talked about these have widespread patterned inputs. We know that they're involved in arousal levels, and I'm going to show you some of the data that links these QPPs to different levels of arousal. We know that if you manipulate different neurotransmitter systems, you can change functional connectivity, which is not surprising. But since these patterns of QPPs actually account for most of the functional connectivity we see, that implies that it can alter them too. And something that we thought was really interesting is that these neuromodulatory systems are some of the sites of the earliest alterations in neurodegenerative disorders. And we had already kind of played around with these in a mouse model of Alzheimer's disease and found that the QPP, the patterns actually gave better classification of the Alzheimer's versus the control than other functional connectivity metrics. So it seems like, maybe these are something that's affected early. Okay, so to go back, and kind of the rationale and why we think these deep brain moduli might be important, I want to show the timing of the QPPs. And I'm showing these in yet another way here. I know it's a little bit confusing. What we're doing here is we're showing the course of the template along the horizontal axis. And then at each time point of the template, we're showing where different areas peak. So for cortex, you have a negative dip mostly at this point, a positive dip mostly at this point. And these black lines are the left posterior cingulate cortex, a major node of the default mode network, when that peaks and when it dips for reference. So we don't need to look in detail at the rest of these. I want you to focus in on the thalamus and the brainstem and deep brain. And if you look at those, you see there are a lot of areas in the thalamus and in the brainstem that peak before the cortical peaks. Like maybe these are drivers. So the timing made us think that, maybe there's something going on with this arousal system. So we did a few other things while we were trying to get to a more concrete experiment. We looked at the Human Connectome Project scans. And these scans are, I think, 10 minutes long, 20 minutes long, 1,200 images, and TR is 700, so whatever that works out to be. So what happens is you put people in the scanner, and a lot of us get drowsy over time. So we wondered, well, maybe there will be a change in QPPs over time. So what we're plotting here is the time into the scan. So this image number along the horizontal axis, and the number of subjects that in which each QPP is dominant at that time. And so QPP1 again was global signal, QPP2 was the default mode network task positive, and then we have the third one. And what we notice is that if you look at this global signal, it tends to increase over time. Whereas the QPP related to the DMN and task positive network tends to decrease over time. And that is consistent with what we would expect if people are getting sleepy and relaxed in the scanner. It's known that as you get drowsy, global signal increases. And because, as I'll show you later, the DMN and TPN pattern has been linked to paying attention, it makes sense that that would be kind of anti-correlated with the global signal. We also looked at the relative distribution of these different patterns in rats that were anesthetized with different agents, dexmedetomidine, which is more of a sedative, isoflurane, which is more of an anesthetic, and then a
Are other folks seeing the paused video?
Yeah, it seems to
Yeah, it's
be
paused for me too.
paused and
done,
everything. Yeah. No longer any noise or sound or anything.
Hey, Shella, can you hear us?
Yeah, I can hear you. Can you guys hear me?
We can now, but
Okay. Do I need to go
not
back?
for
I just saw a thing that
about
my
30
internet
seconds.
was
You want to go back one
unstable.
slide, maybe?
One slide? Okay.
I think so. Is that about right, folks?
Yeah.
Yeah, that's great.
All right. Thanks, Shella.
So this was looking at the distribution of QPPs over the course of the scan and HCP data. And we saw QPP1 increasing at the expense of QPP2 because QPP1 is like global signal. We know global signal increases as people get drowsy. And so this is consistent with people getting drowsy over the course of the scan. And also another link to arousal for these patterns. We looked at the distribution of the same patterns in rats under different conditions and saw that there were differences, although it's kind of hard to interpret what those differences actually mean for a given anesthetic condition. And so then this slide, we did the same thing in data from another group where they had rats that were either sedated or rats that were given sodium pentobarbital to reduce the brain activity to 0. But they're still breathing and their heart's still beating. And what we saw is that we had strong QPPs still in the sedated animals. This is not well labeled, that's the top row. The second row is the isoelectric animals, and most of the structure goes away, which was good evidence to us that we're not somehow just inducing these patterns via respiration or cardiac pulsation. Okay, so going back to the idea of neuromodulation, there's this great map of different receptor patterns from Hansen et al. And one of the cool things is if you look at this map, we can see features that align with some of the features that we see in the resting state functional connectivity. For example, norepinephrine tends to drive
Dead again. No
Yeah.
sound.
The last word she said was epinephrine, I think.
Yeah.
Hey, Shella, the last word you said that we heard was epinephrine.
Okay,
Maybe
okay, I'm sorry. I don't know what's going on today. Usually my internet's pretty good here.
That's okay. But maybe you want to turn off the video it might help, right?
That's a great idea. Oh, that's
The Zoom video. Yeah.
Yeah, I think I'm getting it on the other thing. Here we go. Let's try that.
Yeah, go ahead. Okay.
Okay, so sorry, let me go back. Okay, so these are the patterns of receptors. We talked about, I think you said you heard me say norepinephrine, and
Yep.
those tend to align nicely with somatosensory cortex, and that's one of the areas that shows the earliest changes in most of our QPPs. So that's, very intriguing. Another one, for example, here is acetylcholine is showing a high concentration in that kind of precuneus area that's a key node of the default mode network. So, these kind of match with some of the features that we see in some of these patterns. Again, suggestive, but not conclusive. So we looked into a few other kind of more specific studies. This one was not done by our group, but it came from a group at NIH with Jeff Dunn and David Leopold. And what they did is they took monkeys and they injected something into one side of the basal forebrain at a time to inactivate that area, and then looked at what happened to kind of this global signal. And depending on which side they injected into, they saw a decrease in that side's amplitude of fluctuations in the global signal, which would be corresponding to QPP1. So we have ties then between QPP1 and these basal forebrain areas that they inactivated. So I was part of a collaboration recently, led by a former postdoc of mine, who started looking at this global signal in QPP1. And he found that pattern that we have of activity in that QPP lines up really nicely with similar patterns and a lot of features that arise from the autonomic nervous system. Things like pupil diameter, skin conductance, heart rate variability, respiratory volume, wideband changes in EEG. And so one of the things he's proposing is that maybe this is part of a unified sort of autonomic signal, this whole global signal, basal forebrain, autonomic peripheral system is all kind of one integrated system, which I think is super cool. And there's a lot of work that I want to do to kind of investigate that now. So this is kind of how we're thinking of QPP1. The QPP2 with the task positive network and default mode network, we've been thinking might be linked a little bit more closely to the locus coeruleus input. We know that the LC is involved in task performance and trial-to-trial variability, and people have shown that if you stimulate the LC, you can alter infraflow activity in the cortex. I didn't show you this, but infraflow activity is very closely tied to the propagation of activity that we see in the QPPs. We also did a study where we saw that QPPs were weakened in ADHD, which would be consistent with reduced adrenergic input. And we have animal experiments that show if you give DSP-4, a selective LC neurotoxin, you weaken the QPPs to the point where we can't really detect them. So we wanted to understand this relationship a little bit better. Again, we kind of looked at existing data first. So I mentioned that the LC is involved in task performance. So we looked at QPP1 and task performance. Sorry, that should be QPP2. I told you we have nomenclature issues. That's an old slide. Anyway, we looked at QPPs while people were either resting or doing a psychomotor vigilance task. Basically watching a small dot, occasionally it changes color, and you hit a button as fast as you can. We didn't see any change in how often they came or how strong they were. But if we looked at the phase, which part of the cycle you were in, we saw a difference in reaction time. So if the default mode network was dominant, that's this first half here, we had much higher variability in reaction time than if the task positive network was dominant. So we thought that was an interesting tie to potential LC input. A collaborator of ours looked at QPPs when people were in the zone doing a task and out of the zone doing a task. And we saw changes in the participation of the task positive network in the pattern. So that kind of fit with the story that we were finding so far. And then we did finally the experiment we really wanted to do. And we went back to rats and we worked with a collaborator to do optogenetic stimulation of the LC. Now, we did multiple different types of stimulation because the LC has different firing modes. We used a 2 Hertz for low tonic, which is kind of the relaxed version state, 5 Hertz for higher tonic, which is a little bit more of an anxious state, and then a 15 Hertz phasic stimulation that's more what you see when an animal is engaged in a task. And this turned out to be messier than we expected. But we did see an effect of the virus, so that we saw differences in QPPs in the animals that got the virus and the ones that just got the control injection. We can't tease apart anything about the different stimulation frequencies, and we see some differences even in baseline, which is, like I said, it's messier than we hope. We think what happened is we performed the LC stimulation for 10 minutes at a time, and we did these in randomized order. So even the baseline scans are not necessarily at the very beginning. It just means they got no stimulation. And so we think we kind of overdrove the LC so that we had lingering effects past that 10 minutes. So we would like to do a cleaner experiment for this, but we do think that the effect of the virus shows that the LC stimulation is somehow changing these patterns, so it is likely to be involved. Now, I said it's basal forebrain for QPP1, it's LC for QPP2. I don't think there's really clean separation of these nuclei. I think if anything, we're looking at kind of different aspects of the same system. And, this is shown nicely. These are the patterns you get from stimulating optogenetically different cell types in the basal forebrain. And you can see you can get a lot of different patterns from that. So I think these are very intertwined, and we have a lot to do to kind of figure out where things are going with this. One of the really interesting hypotheses that I've heard is that there are times when the brain activity is more dominated by norepinephrine signaling, and sometimes when it's more dominated by the acetylcholine signaling. So that maybe those two kind of drive the brain between integrated and segregated states. Matt Schein has done some nice work on that. And so one of the things we'd like to do going forward is to do some more optogenetic stimulation work in animals and see if we can kind of confirm that sort of hypothesis. So anyway, here's our working model for these large-scale patterns that we see. We think that they're related to neuromodulatory input. We think there's also an aspect of network structure. So maybe you get periodic neuromodulatory input to certain sites, which then kind of propagates over the structure. We know that if you do a callosotomy, you kind of disrupt the bilateral symmetry of the patterns. There could still be some effects of this similarity of the vascular system. And like I said, we can talk about that later if you want, but this is kind of what we're thinking right now. So the last thing I wanted to mention, we're also very interested about how these spatiotemporal patterns and the sort of wave-like propagation at the large scale can influence the fast local activity that we see in, say, columns or layers or cortical areas. And so one of the things that a former postdoc did was to look at how your response to a short visual stimulation differed depending on where you were in the cycle of one of these QPPs. And what we found is that it accounts for a lot of the variability that you see in the response. And so we got very different responses depending on where we were in that cycle. Okay, so I'm going to wrap up there because I want to leave time for discussion. We have code for all of our pattern finding algorithms available, and we're also happy to help you guys if anybody's interested in getting that set up. And I want to say thanks to the lab and especially Wenju. I talked a lot about our human data today, but Wenju is our genius who makes all of our animal experiments possible. And I'm really excited about some of the work we've got going on there. So thank you for listening.
Thanks, Shella.
Thank you. If you have any questions, you can put it in the chat. But we'll get started with one that's in there about the length of templates. So there's a question, Luis, if you want to come up and discuss, you should be able to unmute yourself.
Yeah, I'm happy to. Hi, Shella.
Oh, hi. I didn't see you on there before.
Can you hear me?
Yeah.
Yeah. So as you might suspect, I was thinking about your QPPs, and if I understand them correctly, it's at a frequency basically the window is fairly long in terms of 20 to 30 seconds. I think that one that you showed, the plot, that some of the regions when they came in and out of phase, and it seemed to last 30 seconds if I understood your explanation. And so, as you know, with the switching linear models, we had found these states that transition quite more rapidly between 3 and 10 seconds. And so we don't have them as repetitive patterns, of course. It's with a different kind of analysis, but based on your work, but your view of the literature more generally with fMRI data, do you think that the more robust patterns or the more noteworthy patterns are these longer half-minute ones, or there might be something that is happening in faster temporal scales that might possibly repeat to some extent, but not perhaps as quasi-periodically?
Yeah, absolutely. I mean, I think that's a great question, and I think that's a very interesting thing that we haven't had a chance to completely explore yet. So one of my postdocs did do some analysis. I guess let me start off by saying first that, yeah, the QPPs are always fairly long, 20 to 30 seconds with fMRI. But that matches very well with the peak frequency that we get in the bold fluctuations, which is about 0.05 hertz. So there's an inherent limitation due to the vasculature for fMRI. Now, I think there's evidence that if we go to optical imaging, and the thing I'm most excited about right now is the simultaneous wide field optical imaging and fMRI we've got going on. If we go to optical imaging, people have showed that you can get not necessarily QPPs, but these sort of motifs at high frequencies and then again recapitulated into the infraslow frequencies. And so I strongly suspect that we're going to be able to extract these same sort of patterns at higher frequencies in the optical data. But I don't think that these are necessarily the only interesting things in resting state fMRI. I think this is some kind of background arousal brain state sort of driver. And then I think there's probably a lot of interesting information overlaid on top of it. It may even be the most interesting information. So one of the things I've been trying to convince NIH to do is to let me look at, if I take out the QPPs, do I get better specificity to tasks, to individuals, things like that, with what's remaining. So is the interesting stuff in the QPP or the leftovers?
Right. But there might be complex combinations of them, right? Like you might difficult to maybe disentangle.
Yeah, I think that's
super clean data and whatnot. Yeah, great. Yeah. Thank you.
Hi, Shella. Quick question about the QPP3. You kind of mentioned it briefly, but you didn't want to go into details during the talk. Do you think, could you give us some idea of what you think it might be going on in the QPP3?
That's a great question. I don't know. I know that it matches with the gradients. I didn't talk about this today, but Taylor's paper, when he used the CPCA algorithm, shows that our QPPs match up nicely to those functional connectivity gradients that people sometimes talk about. So it's as if the gradient arises from these patterns that sweep across the brain. Or I guess, if you want to think of it another way, you could say the waves sweep across the gradients, right? I don't know which one comes first. And QPP3 is consistent with that third gradient that they see. But nobody has really done a lot of research into where that comes from or why it is how it is. So I think that one has been kind of neglected so far. We've spent a lot of time trying to understand the other two over the last few years, but the third one accounts for less variability. And one of the things that's really interesting, and I think makes this an important area to look at further, is I just saw a presentation in NUSA by Ting Xu, who's looking at these sort of patterns in children as a function of development. And they actually swap which one dominates over time. And so, maybe that third pattern is more important when we're very young and still, kind of solidifying everything, and then becomes less important as we get older. I don't know, but I think it's a very interesting area to explore.
Cool. Thank you.
Shella, I wonder if you could say more. I'm just thinking about the time scale of these patterns and your effects, and you showed these reaction time results earlier. We have seen, it was only in a preprint, wasn't never fully finished, but we saw these reaction time effects for faster oscillations in a task that I feel in a working memory task in relation to things like 10 hertz theta alpha oscillations. Your signals are, of course, slower than that. And I just wondered if you but then also you were talking about optical patterns. So we're talking about at least 3 different time scales of things. And I wonder if you have any thoughts about how to put it all together, because there's a lot of things that are all real, but it's hard to put the big story together.
So we're trying to do that now. I agree. I think this is a fascinating question. And I think it's something that we haven't investigated at all really yet, is how brain activity transfers information across scales. Because I think it goes both ways, right? I mean, you can think of the localized activity as giving rise to the global state, but that global state also places some constraints upon that localized activity.
Mm-hmm.
And so what we're doing right now, as I mentioned, we've got the simultaneous wide field optical and fMRI. And we have only begun to start to look at this, but we have a project going on to see if coactivation patterns in the optical imaging are aligned with these quasi-periodic patterns in the fMRI. Like maybe there's some sort of cyclical variation in probability that one occurs.
Something like phase coupling.
Yes.
Maybe?
Yes,
Is that asking for too
exactly.
much? I don't know.
Maybe it is. Maybe it's not. I don't know. I think it's worth checking into. And we did see. I have results from one mouse. This is why I'm not showing it today. But we did see that if you look at the one mouse, there are certain times. Each coactivation pattern tends to localize to a certain phase of the QPP. Not, exclusively.
Oh, that's interesting. Oh, it's exactly what I was kind of thinking about. Yeah, okay.
Yeah, so I hope that will continue to show up, but I think trying to understand how things carry from one domain to the other is really interesting.
Because if something like that works, then it's in the direction of there being like a hierarchy of these rhythms, right?
Well, and I mean, I kind of think of it that way, and part of this is based on that optical study, right? This is Mohajerani's group, where they did the patterns, I think, between 3 to 6 hertz, mapping back to the same sort of patterns in infraslow, just it's kind of slower frequencies. And, I think that could happen with the bold signal as well, right? So, we could get to even lower, some of these high frequencies may still carry over to give us information in those lower frequencies. And one of the things I did to convince myself that this is true is I did a MATLAB simulation of a traveling wave at like 10 hertz and then sampled it slow. And you can get a traveling wave over the same areas at, 0.1 hertz. So
You mean through some kind of aliasing or
through
something?
some kind of aliasing.
Yeah.
So, I mean, trying to disentangle all this is hard, and especially, with fMRI, we've got the extra filtering of the vascular system.
Right.
But I think it's plausible that information is still there in some form.
Right. When you start talking about the vascular, the kind of filtering, it's hard to know if what we're seeing at this frequency is the actual signal itself, or if it's some kind of aliasing or something like that. Yeah.
So I will say, with the optical data, one of our collaborators has gotten QPPs out of the optical signal, and so that's fluorescence, should not be contaminated with the vascular delay or anything. She has not yet looked at kind of these in a frequency dependent manner. She just looked for the infraslow. So I don't know if she's gotten the same thing if you go to higher frequencies. So I'm very curious about that. And we have just finished our first analysis of wide field optical data. And one of the things we can do is measure fluorescence, hemodynamics, and then fMRI all at the same time. And what really astonished me is that the infraslow fluorescence activity is very tightly coupled to hemodynamics. So that seems to be real, right? That there's something real in these slow fluctuations in the neural activity that the hemodynamics are picking up on. I don't know how exactly it relates to higher frequency activity, but there's some connection there that we just need to parse.
Thank you. Ganendra has a question.
Yeah, and really interesting talk. Like I have two questions. In first one, you showed that these networks are formed from, let's say, from a small, faster region to a larger one. So I was wondering, did you found any patterns or let's say any correlation between these two? That and is there any way like how these large network of patterns are being formed, all the which are operating at a slow time scales, and the slow ones, the small patterns operating at a fast time scales? Did you found any observation between those?
No, again, I think that's something that's very interesting, but we're just getting the tools to really investigate that. So one of the things that the optical imaging will do is to let us have higher spatial and temporal resolution along with the fMRI for the same areas, so we can compare those. With fMRI, people have looked at things like columns and layers, but then they usually don't do whole brain or they don't do fast sampling. And so it's hard to kind of link that activity to these kind of large scale patterns. So I think that's a great thing that needs to be investigated, but I don't know that anyone has actually looked into it completely yet.
Just to follow up, like, is there any differences occurring when we do measurements at different layers of the cortical, let's say, cortical grid, let's say at layer 2 and 3, layer 5, or is there any specific differences being observed?
That's a great question. In terms of resting state functional connectivity, they do see some layer-dependent differences. They are linking those to inputs and outputs in some cases. It's messy again because it's filtered by hemodynamics and the vascular density is different for different layers. So it's a little bit tricky to fully, disentangle that. Nobody has done that yet with optical that I know of, because with the wide field optical, we mostly get just the cortical surface. So layer 2, 3. What I can say though, is that when we look at that cortical surface versus the deeper signal that we get with fMRI, it seems to be pretty much the same.
Right. My second question is for the hemodynamics, especially there are differences at the arteries and capillaries because both different kinds of cells are interacting there, neurons and glial cells. So is there any differences you observe, especially for, let's say, neuronal activity, or let's say if you're measuring calcium activity from glial cells? So you mentioned about hemodynamics. So I was wondering whether is there differences at the because these basically these vasculature go from top to bottom and then they bifurcate into different branches, and majority of the transmission happen at these capillaries. Or I was wondering whether how much differences is being observed overall in terms of, let's say, cortical and then these hemodynamic signals.
That is a tricky question. There's a lot of research on it. There are different findings based on animal condition and where they look. I mean, definitely people see, this arterial control of dilation. They see interesting things with capillaries. Sometimes capillaries are recruited. Some capillaries don't seem to respond in any organized manner when there's stimulation. And then, the venules tend to be more of the source of the MRI signal because that's where the deoxygenated blood collects. That said, I mean, are there major differences at the level of, say, an fMRI voxel? I think no. So besides doing bold fMRI, where we're looking at deoxygenated hemoglobin, we can also do cerebral blood volume, and the patterns look almost the same. We can do perfusion with arterial spin labeling, and the patterns look almost the same. So all of our hemodynamic measurements seem to give us more or less the same thing.
Interesting. I mean, because what I've looked into literature also, that these bold signals are not purely neuronal in nature. even though these glial cells also have an important role on these bold signals. So
Yeah.
that's why I was wondering that how much contribution is there? Like, do you see any differences in fast neuronal activity? Those are excitable cells, and calcium activity, those are non-excitable cells. So that is why I was asking you this.
I think we're still trying to figure it out. I mean, most of the tools to answer those kind of questions have just been developed in the last few years in terms of doing it with fMRI. I mean, if you do it with some other kind of measurement, sure, people have looked at these different types of activity, but there's not as much linking it to the hemodynamic response that we detect with fMRI. So there's kind of a gap that people are trying to fill either by inference or by going in now and actually doing things where we optogenetically stimulate different types of activity and see what the responses are.
All right. Yeah. Thanks a lot for elaboration. Enjoyed the talk. Yeah.
Thank you.
We have another question in the chat from Rodrigo. You can go ahead and unmute and ask yourself.
Thank you for a really fascinating talk, Shayla. I'm definitely convinced that these things are very important. My question is, how confident are you that the templates that you use to detect the QPPs? Because as I understand it, you're defining them based on other data, or and then you're applying it to each new data set. So do you think that there are other patterns that are not so dominant that are being missed that would be useful to study? And do you have any suspicions about what those might relate to functionally or physiologically?
Yeah. Sorry. So let me clarify first. The templates, I did show one that was done on group data, but we don't usually apply that back to individual data. Usually we calculate the template itself for every individual or every group. And we have a paper out, it's Yusefi, who was the lead author on it, where she looked at every individual template from Human Connectome Project data. And they're pretty reliable at the single subject level. Now, are we picking out all of the interesting details? Are we seeing interesting differences in patterns? I don't think we've even tried yet. I mean, we've barely begun to try to see how QPP is different across, say, populations, much less across individuals. We need better statistical tools for one thing. It's hard to take these patterns and say, oh, well, this is significantly different at this area at this time. And so we're trying to think of better ways to do those types of comparisons. And I would love to hear if you have ideas.
I'll think about it. Yeah, I would love to chat with you another time then, once I've had time to think about it. But
Awesome.
I do think there's something to the time varying nature of these things. That's why the static functional connectivity misses them. But even within the framework that your framework is clearly better at capturing things that are varying, but there could be even more variance that it would be really interesting to study.
I completely agree.
I think that's a really interesting question. I mean, I just think that there is this possibility that there's so many more spatial temporal patterns, waves, and other things in the data that we just don't understand yet. It seems like the more we look for, the more we see.
Yeah.
And it's not a false positive. I don't believe they're false positives at all. I just think it's really interesting and complex.
Yeah, I 100% agree. And I think, part of the question is, how do you find them in some sort of unsupervised way? How do you compare across individuals? One of the things that I hate is that we take our beautiful brain data and we smear it and we stretch it and we distort it to register it to some kind of common atlas. I mean, we need to do better than that, especially I think if you want to see the fine details, I think that really makes a difference.
Yeah, in our human recordings, we see a lot of things for individual people that if we co-registered across all people so that every person's brain was the same, we would miss these things if we averaged. And so I feel you. I'm with you. I don't have a solution, though. Any other questions?
I have one more question, if that's okay.
Yeah, go ahead.
I had a question about the quasi-periodic nature of these signals. So now clearly they're not perfectly periodic, right? It's a brain. But I was curious to what degree your analysis is sensitive to slight deviations from the periodic pattern. And the second question is like, what is actually causing these deviations from the periodic pattern? Do you think the entire thing is just stretched out a bit more, or are there certain stable states within the pattern that themselves become stretched and compressed?
Another great question. You guys have all of the best questions. I wonder about all of these things, and I just need more people to help me figure it out. I suspect that they are pseudo-stable states at kind of the positive and negative phases. And I suspect those are the ones that get stretched. The transitions seem to happen very quickly and stereotypically in between those states. We have not seen a lot of variation in the pattern length. You know, I say 20 seconds, I'd say maybe plus or minus 2 seconds in the population.
Yeah, yeah.
But one of the things I like about the complex PCA approach to extracting the patterns is that you don't have to set a length. And so you can actually pull out the patterns and then look at the length that you get, which is a little bit different than saying, oh, I'm going to look for things that are 20 seconds long. And we get more or less the same results using that approach. So I don't think the variability is that high, but I think it might be interesting.
Cool. Thanks.
Irfan, Uma?
I guess if there are no other questions, we can wrap up.
Well, Shella, thanks so much. This is my favorite kind of talk in terms of it opening new directions and new things to think about. So thank you.
Well, thank you for having me, and I'm happy to talk with anyone who has good ideas about how to go forward. I think you've done an excellent job of showing us exactly how much is still missing from this type of research. So
Cool. Well, thanks again for your time. Thanks for everyone for coming. Please join us next month. And for anyone who's interested, this will be posted online so we can have a record of things.
all right, thank you
Have a great day, everybody. Thanks again, Shella.